right, today we are going to look at our Intro to Genetics notes. So we're going to be studying genes, variation, heredity, and living organisms. So in order to fully understand genetics today, we're going to be pretty much starting with the basis. So key terms we need to know, how do we first find genetics, and the basic Punnett square. So before I begin, I want to cover some important vocabulary. So if you ever hear me say true or pure breeding, that means the offspring is always going to have the same trait as the parent no matter what. So if I was taking a purebred golden retriever and crossing it with another purebred golden retriever, I know exactly what genetics I'm getting in the offspring. That puppy is going to match the parents and also be a purebred golden retriever. Self-pollination is when a plant fertilizes itself, whereas cross-pollination is one plant fertilizing another. So today we're going to be looking at Gregor Mendel, the father of genetics, and he studied plants, so we have to understand self-pollination versus cross-pollinations. And then we'll be also looking at different generations. So let's say I was studying your family, and the first generation I started to study were your grandparents. So your grandparents would be the P generation, or the parent generation. Then their kids would be the F1 generation. So your parents is the F1 generation. You would be the F2 generation. If you had kids, they'd be the F3 generation. If your kids had kids, they'd be the F4 generation. So kind of getting where I'm going towards, I could count on and on and on all the way down to F50 or F500. So it's just looking at the sequence of generations that I am studying. Some other important vocab, genotype, so that's the genetic combination of alleles. So homozygous dominant would be big T, heterozygous, big T, little t, or homozygous recessive, little t. So when I'm talking about the genotypes, we're looking at those letters. If I talk about phenotype, think phenophysical. I'm going to be looking at the physical characteristic that the genotype codes for. So you're going to be doing practice later on today to prove to me you understand genotype and phenotype. So geno, the alleles, phenotype, the physical characteristic. So with our genotypes, we could see different combinations, as I said earlier. So homozygous means both alleles are the same. If you think of the root word homo, it means same. So homozygous dominant or homozygous recessive. So this would be that true breeding or that pure breeding we're talking about. Heterozygous, hetero means different. So the alleles are different. So you'll have one big T, one little t. And then hybrid is the offspring of crosses of parents with different traits. So for example, I could take a golden retriever and a poodle and I'd breed them. So I'd get a golden doodle. So it's going to be two purebred parents creating that hybrid in the middle. So to start off our history of genetics, we have this guy, Gregor Mendel, who's really important, considered the father of genetics. So he identified all the different rules in genetics, heredity. He saw how traits were passed down from one generation to the next. So he was actually a monk, and he studied peas in his garden. So he was responsible for tending the garden. He was fascinated by the peas. He saw all these different characteristics. We could see round versus wrinkled seeds, yellow and green, purple and white flowers. So you can see there's 1, 2, 3, 4, 5, 6, 7 different characteristics that he studied with his peas. So in his experiment, what Gregor Mendel did is he took 2 true or purebred pea plants, so maybe a purple flowered pea plant and a white flowered pea plant, and he cross-pollinated these. So usually this is done by bees or wind. So what he did back in the day, he took scissors and a paintbrush, and he just took the pollen from one plant and put it on the other in order to fertilize those flowers. So first example, he took a purebred tall plant and a purebred short plant. So thinking about what would happen, think about your hypothesis right now. Will it be all tall, all short, medium pea plants? What would happen when he crossed these? So what he found is that the F1 generation was all tall, which just kind of shocked Mendel. So he expected our traits to blend. Why do tall and short not make medium? Why crossing a tall and a short plant did all the children be tall plants? So what he decided to do, he's like, all right, I'm going to self-pollinate these plants. So I have my F1 generation, all tall plants. Well, what happens when I cross the 2 tall plants together? So what he found was that he would get this 3-to-1 ratio. So 3 plants were tall for every 1 short plant. So he repeated this experiment over and over and over again, looking at different characteristic traits besides the height. So we looked at flower color of the seeds, the wrinkle shape. And what he noticed is that this F2 generation was always a 3-to-1 ratio. So what he kind of figured out is that traits can be dominant or recessive. So here's just looking at examples. I have my purple and white flowers, F1 generation, all purple, F2 generation, that 3-to-1. Pea plants, or the pea color, sorry, all yellow, all green, found in the F1 generation, all yellow, F2, that 3-to-1 again. So Mendel's conclusion, he realized that the inheritance of traits are controlled by genes contributed from each parent. So gene, that should be a vocab term we already know, a small segment of DNA. So we know that gene can turn on or off in order to code for a trait. So if I turn my gene on, it's going to be transcribed into mRNA. That mRNA will go to the ribosome to be translated into a protein, and that protein is expressed into your physical trait. So these genes came in different forms called alleles, so that might be a new vocab term for us. What an allele is just a different form of a gene. So here I can see my homologous pair of chromosomes, so homo meaning the same. So if I have homologous pairs of chromosomes, they're going to be the same chromosomes coding for the same traits. So here we can see this chromosome is coding for white flowers. This chromosome is coding for purple flowers. So these would be examples of alleles. So they're coding for the same trait, the color of the flower, but there might be different forms. So I could have an allele that codes for purple versus an allele that codes for white. So what he realized is that some alleles were dominant, meaning they would always be expressed and they would cover up that quieter recessive allele. And then if you had two lowercase recessive alleles, that's the only time the recessive trait would be shown. So 3 different things he worked for and found is 1, the rule of dominance, so which allele will be expressed, the dominant or the recessive, the law of segregation, and the law of independent assortment. So we're going to look at all 3 of those here quickly. So the rule of dominance, one trait hides or masks the other. If a dominant allele is present, then the recessive is masked. So here I can see this is a Punnett square that we're going to be practicing with later. So on each side of the Punnett square, we're going to have the haploid cells, so like the sperm and the egg cells. So here I can see I have 2 heterozygous parents, big G, little g, and you can see the cross. So the big G's drop down, this G goes over, this G goes over, and then the 2 little g's. So here I can see when I cross heterozygous parents, that offspring is going to be a 3-to-1 ratio. So for every set of offsprings I have a 75% chance of it being green and only a 25% of the recessive trait showing, which is white. So with simple dominance, which we're going to learn other types of dominance later, so simple dominance, if I have that capital letter, it's going to mask or cover the recessive lowercase allele. The law of segregation, we talked about this a little bit in meiosis. So this just says that allele pairs will separate during gamete or sperm egg formation and then randomly used to unite. So here I can see my female parent, she's heterozygous. So one big T will go to this egg, the little t will go to that egg. Here's my male, big T, little t. So the dominant T to one sperm, the lowercase t to this sperm. So depending on which egg and sperm get fertilized, that's the chances of the offspring. And then the law of independent assortment, this just states that when 2 or more characteristics are inherited, that the combinations of the trait in the offspring don't always match the combination of traits in the parents. So just because my dad has brown hair and blue eyes doesn't necessarily mean that I'm going to have brown hair and blue eyes. So the below section is my family. So my dad has brown hair, blue eyes. My mom has brown hair, hazel eyes. I have brown hair, green eyes. So just because I inherited my dad's brown hair doesn't mean I necessarily got his blue eyes. So the law of independent assortment is saying that I could get one trait from one parent and another trait from another parent. So our genetics, our traits are independently assorted from one another. So today we're going to learn our first pattern of inheritance, which is just the basic Punnett square. So I'm hoping you had experience with this in middle school, and this should just be a refresh on simple dominance. So here I can see the gametes go on one side. So let's say these are the egg cells, these are the sperm cells, and we're going to fill in that Punnett square to see the chances of each offspring. So the Punnett square is named after Reginald Punnett, and basically it's a box that shows how the sex cells combine to make the zygote or the fertilized egg. So we're actually just going to practice this together. So practice problem 1. In pea plants, purple flower color is dominant to white flower color. A purple flowered plant is crossed with a white flowered plant. So our first thing is to determine the genotypes of the parents. Well, if I know purple is dominant, I'm going to assume the parent is either homozygous PP, so big P, or they could be heterozygous, big P, little p. The other parent is a white flower. White is the recessive, so I know no matter what, that parent has to be little p, little p. So you always follow the letters based off of the dominant trait. So if the dominant trait is purple, it's big P, and then white would be shown as a little p. So determine what percent of the offspring could be white. So pause the video now, see if you can set up this Punnett square on your own, and then I will go over the answers. So here's our answer to Punnett square problem 1. So here I have my homozygous purple parent, big P, heterozygous white parent, little p. So I can see 100% of the offspring are that heterozygous purple. Practice problem 2. In humans, brown eyes are dominant to blue. If a man with blue eyes, so let's think, I know he has to be little b, has a child with a woman who has brown eyes, so she has to have at least one big B, but her mom had blue eyes. So you might have to set this back to figure out what the mom was. But I know she's going to be heterozygous because if her mom had blue eyes, that means she has to receive one of those lowercase b's from her mother. So what's the percent chance of them having a child with brown eyes, and what's the percent chance of them having a child with blue eyes? Pause the video, see if you can solve this problem. All right, and here's our answer. So I can see this is the woman. She had the brown eyes shown, but she received this little b allele from her mother with blue eyes. And then I know dad had blue eyes, so he's homozygous recessive. So I have a 50% chance of the offspring having brown eyes and a 50% chance of the offspring having blue eyes. So that's our basic Mendelian notes.